Hello and good afternoon. We are back with our final speaker of the evening, and I would request Professor Dr. Arup Kumar Mitro to introduce our speaker. On to you, sir.
Thank you. Thank you, Moitri. It is really my privilege to welcome Chandrabali Dutta as the ultimate speaker for the Frontiers in Biological Sciences. This particular program we thought of initiating almost a year back, but never really we had any idea that it will be a webinar, but somehow we have to change with the scenario, with the new normal. We need to refresh ourselves with the technology. Now, when it comes to technology, naturally there will be hitches and we have to cope with it. So it is really my privilege to introduce Dr. Chandrabali Dutta, who was my student way back. I think it was in 2003-4, long time back. And I really am feeling proud in this platform to introduce your own student who has reached a new height in her career. She completed BTech in Biotechnology from West Bengal University of Technology, and then subsequently pushed off to University of Leeds for doing her master's in biotechnology. Then she completed the degree and worked as quality control manager and assurance in Didcot, Oxfordshire. She had almost 6 years in R&D in GlaxoSmithKline, Hertfordshire, and presently she is working as quality assurance manager at the clinical biomanufacturing facility of University of Oxford. And incidentally, she belongs to the same group working for the development of COVID-19 vaccine. And we can see that she has spent her research career in industry. So it will be a totally new aspect of learning and knowledge. And this time it is quality assurance in vaccine production, which is the title of her talk. So I really believe that people listening to her lecture will be immensely benefited from her talk. So over to Chandrabali for her lecture. Thank you very much.
Thank you, Arup sir, for giving such a great introduction to me. I feel honored to St. Xavier's College Microbiology Department for inviting me for this current scenario on COVID-19 talk series. And I'm not the last but not the least speaker in this session. And it's a pleasure and honor to be part of this event. And as sir already mentioned, I haven't done a PhD, so I think people got confused. They call me Dr. Chandrabali Dutta. I feel really honored, but I'm actually not a doctor. I've done a master's in biotechnology. So my experience is from industrial background, and I'd like to share some of the current scenarios which are going on and talk about the vaccine, obviously. I'd like to confirm if my screen is visible to everyone.
Yes, ma'am, it's visible.
Okay, thank you. So, so my talk today mainly highlights on aspects of quality assurance in vaccine manufacturing. So just introducing myself, I'm Chandra. I work as a quality assurance manager at the clinical biomanufacturing facility within University of Oxford. And already I thanked Arup sir for inviting me for this presentation. And I'd like to share some current scenarios, what's happening in UK with the vaccine and what's happening. I'm being part of this organization where we have worked day and night to make this vaccine successful to go for clinical trials. So my talk will mainly cover vaccine update, how did vaccines originate, quality assurance challenges in vaccine production, and role of MHRA, which is a regulatory body which helps us in certifying a vaccine to go for clinical trial, and obviously good manufacturing practice, which is a requirement for a clinical trial organization to follow MHRA guidelines before they can go to the clinical trials, and a short experience of my journey to this stage at this moment. So I'm sharing a little video from BBC because due to confidentiality purposes, I cannot share everything which is within the organization, but I'm obviously allowed to share which is common knowledge and is distributed within public at the moment. So just to make my talk a bit interesting, rather than going through presentations and slides and talking, I thought I'd add a little video to make it more interactive and give a current update what's going on here.
The first human trials in Europe of a coronavirus vaccine has begun in Oxford in what is a highly significant moment. This afternoon, 2 volunteers, both scientists, were injected. They're the first of more than 800 adults aged between 18 and 55 who've been recruited for the study. Half the group will receive the COVID-19 vaccine and half will get a control vaccine which protects against meningitis. The results won't be known for months. Already more than 186,000 people have died because of coronavirus worldwide. In the last 24 hours, 616 more deaths were recorded in hospitals across the UK. It brings the official UK death toll to 18,738, but that does not include deaths in care homes or in the community in England and Northern Ireland. Here's our medical correspondent, Fergus Walsh.
This could change everything. A vaccine against coronavirus. A way out for all of us.
Okay, so I need a scratch.
The first volunteer to receive it was Elisa Granato, a microbiologist. She's 32 today.
I'm a scientist, so of course I want to try and support science, the scientific process, whenever I can. And since I don't study viruses, I felt a bit useless these days, so I felt like this is a very easy way for me to support the cause. Yeah, that's why I'm here, and I'm excited.
Half those on the trial will get the coronavirus vaccine, half a different jab. The volunteers don't know which one they've received.
I'm just basically going to sit here and incubate this thing and hopefully provide some good follow-up data, and then we'll see, and hopefully it works.
What would normally take years has been done in 3 months by scientists at Oxford University. Around 80 groups worldwide are developing coronavirus vaccines. A few others in the US and China have also started human trials. There's no guarantee any of them will succeed.
Personally, I'm very optimistic it's going to work. Formally, we are testing it in an efficacy study. There's absolutely no suggestion we're going to start using this vaccine in a wider population before we've demonstrated that it actually works and stops getting people infected with coronavirus.
So how does the vaccine work? Scientists have taken the genes for the spike protein on the surface of coronavirus and put them into a harmless virus. This forms the vaccine. After it's injected, the vaccine enters cells which start to produce the coronavirus spike protein. This prompts the immune system to produce antibodies and activate killer T cells, which should recognize the coronavirus in future and destroy it, preventing infection. The scientists will only know how effective the vaccine is if lots of volunteers are exposed to coronavirus.
Well, at this moment, we're chasing the end of this current epidemic wave. And of course, if we don't catch that, we won't be able to tell whether the vaccine works in the next few months. But we do expect that there will be more cases in the future because this virus hasn't gone away.
Why not simply infect people with coronavirus after they've had the vaccine? Then you'd know.
At this moment, it would be very difficult to do that with a disease which potentially has quite a high fatality. So I think if we reached a point where we had some treatments for the disease and we could guarantee the safety of volunteers, that would be a very good way of testing a vaccine.
The vaccine is stored in this freezer at -80 degrees. Rarely has a medical trial had so much riding on it for so many people. It is hard to overstate just how important this vaccine could be if, and it's a big if, it proves to be safe and effective. It would represent science giving us the solution to the coronavirus pandemic.
Well, I think everybody agrees it's the only way we're going to get out of the lockdown, the social distancing, and really be able to still have people protected as they go about their daily lives.
Edward, a cancer researcher, was next up. There is a theoretical risk the vaccine could make a coronavirus infection worse, but the team here think that highly unlikely, and the volunteers will be carefully monitored.
It seems like the right thing to do to ensure that we can combat this disease and get over it a lot faster.
Do you have any concerns at all about safety?
I think you can never fully exclude any sort of potential risk, but I think you have to, I guess, walk in faith in these things. You have to trust in that the work has been done as best they can, and know that the cause is important.
The Oxford team are hoping to have 1 million doses of vaccine ready by September, with a huge scale-up in manufacturing after that. It's not clear, though, who would be prioritised to get the vaccine first. And for now, we have to wait and see if it works.
And Fergus is with me now. Extraordinary how quickly they have turned.
So as you could see, I really wanted to share this video because this is the organization where I work, and the -80-degree freezer where the vaccine I've handled myself for different occasions for labeling purposes. And you could see in the lab, the R&D team was working on the vaccine, and the production quality control checks were being done on the vials to see whether the volume checks are correct. So the volumes are correct. So I thought I could share this to give you a visual presentation where I work and what's going on. So this news was live in near April when the clinical trial started. So, and then the vaccines went from our manufacturing unit to the clinical trials over the road within Jenner Institute. So it has been an enormous team effort, 25 of us working day and night to make this possible. Usually a vaccine development and production to the clinical trial takes more than 3-4 years, which we have actually achieved in less than 2 months. So 1 month in R&D, and then it came to us within the manufacturing unit. So our tech transfer team took the project from R&D scientists and developed it within the R&D team, and then it went to the clean rooms facility where it got manufactured. And me being the quality assurance manager, I was involved in every step of this whole process, making sure that we are compliant, we are following quality procedures, where there's no errors, there's no issues. So this has been a challenging task for us. Everyone has been working under enormous stress because that time lockdown happened and government announced that you can only work from home. But every one of us took our life in danger to going to work and work on this project so that we can save the world, save lives. So I feel really honored to be a part of this team, and I'm going to talk more about how these vaccines originate, what is the history behind vaccines, just to give students an idea how did vaccines originate. So my next video covers the origination of vaccines.
This is the smallpox virus. It infected the Pharaohs, caused a terrible impact on the Greek and Roman empires, and decimated Native American populations. Nothing seemed to stop this lethal plague until the end of the 18th century. At that time, smallpox was a major global endemic disease. Only in Europe, it killed around 400,000 people per year. It didn't differentiate between rich and poor. Even 5 kings were killed. But for some reason, milkmaids and shepherdesses seemed to be immune to the disease. In fact, they were renowned for their beauty because their skins were free of the scars smallpox caused. They were attractive and resistant. But why? That's what the rural doctor Edward Jenner was trying to understand. He knew that milkmaids got small blisters on their hands when they milked cows suffering from cowpox, a mild disease that cattle suffered from, which caused similar lesions to smallpox. And this phenomenon made Jenner think that maybe cowpox and smallpox were related, and that infection with the milder disease would be enough to protect people against the lethal one. As a man of science, he decided to prove it. And to do this, he needed three things: cowpox that he extracted from a milkmaid, fresh smallpox from a patient, and a human guinea pig. He chose his gardener's son, an 8-year-old boy called James FIBS. He infected the child with cowpox, waited a couple of months, and injected him with smallpox. And James didn't become ill. He repeated the experiment with other patients with the same results. Jenner was right, even though he didn't fully understand how his remedy worked. The most remarkable thing was that his discoveries were made way before Jacob Henle proposed in 1840 that infectious diseases were caused by germs, a theory demonstrated by Robert Koch. Decades later, Louis Pasteur, inspired by Jenner's experiments, showed that attenuated microbes could be used to prevent diseases, and he proposed the terminus vaccine in his honor. Almost 200 years after Jenner's experiments, and thanks to global vaccination campaigns, in 1980, the World Health Organization announced one of humanity's biggest achievements: the complete eradication of smallpox, the most deadly infectious disease in history.
So I wanted to share this video with, especially for students who want to understand how did vaccines originate. And when my technical skills are not really vaccinology or virology or immunology, I have actually worked in big pharmas where I was developing inhalers. So when I applied for this job, I was researching on how do vaccines originate? Why did it? Who discovered it? So I was really curious to know. And that time I came across this video, and I thought it would be really good for students to understand how did vaccines originate. And there's so many vaccines all over the world now, to like polio vaccine and rabies vaccine, Zika virus vaccine, Ebola vaccine. So this is really interesting to know that how did vaccines originate. And our institute is called Jenner Institute after Edward Jenner. So my next slide is mainly talking about quality assurance in vaccine production, which is currently my job. So our facility within University of Oxford, we are a bridge between academia and clinical trials. So we function as a mini manufacturing plant where we make sure that every quality assurance is ensured, and we have a quality management system according to European regulatory requirements of good manufacturing practices. So what is good manufacturing practice? What is GMP? As you can see, there's a picture of a book here saying MHRA, Medicines Health and Regulatory Agency, rules and regulations and guidance for pharmaceutical manufacturers and distributors. So this is called the Orange Guide. So this is mainly our Bible. So being quality assurance and a part of my team, this is our job responsibility to make sure each and every clause maintained within this thick book is applied and implemented, and we are following them strictly. Otherwise, we will have a good manufacturing practice breach. MHRA can take our license. So it is a very critical and responsible job, because end of the supply chain, before the qualified professional does the batch certification, it's me who is signing every document to confirm that I'm happy with whatever production has done, whatever QC, quality control department has done. So it's a really responsible and stressful job to make sure that the vaccines which is going into the healthy volunteers for clinical trials is safe, patient safe, and compliant. So in order to do that, we have a quality management system in place. So the core processes within our quality management system are mainly, the first and foremost is the standard operating procedures, forms, and policies. So we have to predefine every procedure or method we follow in the pre-GMP team, which is the R&D phase, to the final production within the clean rooms. So we cannot do any step without following our procedures. And if we do not follow our procedures, we have to make sure we have to do a thorough investigation and find out the root cause of the problem. And there are many forms as well. So each and every raw data is also captured in those forms. So anything you write down, any operator writes down, has to maintain good manufacturing practice procedures. So for example, if an operator is doing an experiment on a Monday and he goes back and starts to write that report on a Friday, then it's not compliant for us because it's not concurrent. So in good manufacturing practice and quality assurance, there's a phrase called, if you do not write down, it is a rumor. So basically, how can quality assurance say that this is concurrent and true data? So basically, everything has to be recorded. It's very tedious when I've been a scientist in the lab in a GMP facility, where when I first came from academia to industry, I was questioning everything. Why do you have to record this? But I was trained to be like that. So for example, I'm using pipette 5000. This is the serial number. This is the calibration date. Everything has to be recorded. So traceability has to maintain, because if something goes wrong, then we can do a thorough investigation and find the root cause. And management of change. So if you want to change a procedure or a simple step within your experiment, you cannot do that without quality assurance approving your change. And then we have audits. So internally, we have internal audit team. We audit each and every department to make sure that we are all following our procedures. And that way we make sure that our organization is compliant before an external party like MHRA comes to audit us. So we have to act as inspectors, and really the MHRA auditors are like inspectors. When they come in, they can ask for anything. And if we do not comply with their requirements, they have the authority to take our license to manufacture. So it's a responsible task for us to make sure we are following all this. And next comes the complaints procedure, where if anything goes wrong in a trial unit, then the trials team will contact us and say, this has gone wrong, please, we need to raise a complaint. And obviously staff training. So any staff working in R&D, in quality control, in manufacturing, have to be trained and certified in a current procedure before even doing that activity. And also validation and verification of assays. So when, for example, if you're doing an experiment, you cannot just say, I've written the SOP and I'm going to do that. No, we have to validate to show that the experiment says the specification for a product is XX. So we have to validate and show that it's actually that, and then finalize our procedures. And obviously equipment and software. So any software we use to use an equipment, for example, spectrophotometer, or in a different pharmaceutical company like HPLCs, high performance liquid chromatography. So you have to have a software to use the, manipulate the data and to analyze the data. For that also, that software also needs to be validated. And then comes out of specification. So if anything goes wrong in production and we do not meet specification as our predefined specification, then we need to do a proper root cause investigation. And deviations, so we have planned deviations in place. So when this COVID project started, we raised a planned deviation, because if we had to write everything and record everything from scratch, then we didn't have time to do in 1 month. So we raised a planned deviation and recorded anything we were deviating, and a risk assessment was added by qualified professionals to make sure that we are compliant. Quality risk management is one of the key things, where we risk assess anything we are doing within our procedures to make sure that end of the supply chain is our healthy volunteer or patient who is safe. Quality event system, we have a system in place to capture any odd events happening within production. So if production has done something wrong, they raise a quality event. And then quality assurance analyzes the scenario and then comes up with lots of questions and risk assessment to make sure that nothing is missed or nothing is not compliant. corrective and preventive actions. So out of all these deviations happening, we raise a corrective action to stop this from happening. Corrective actions immediately to stop this, and preventive actions so that in future we do not do this mistake again. And there are policies in place, steps in place to stop us from doing this. Good documentation practice, as I mentioned earlier, is like recording everything following ALCOA principle, attributable, concurrence, legible, and accurate. And then comes the product recall. So if anything has gone wrong, then we have to recall the product from the trials unit if we find that there's something wrong. So all these tasks are, we have SOPs, mountains of paperwork to go through because we are a paper-based system, and everything needs to be signed and dated on the same day and concurrently. We cannot do any retrospective signatures. If there's a retrospective signature, if someone has missed it, we need to also justify why did we miss it. So it's a really traceable job where you have to make sure everything is compliant. So I have got a little video for mainly students who want to pursue a career in industry and get a bit of a flavor that what are the rules of GMP? Like how does it impact in a live current working scenario where how do you imply those rules? So just some rules and principles of GMP in the next video.
The 10 rules of GMP. Confirm you are trained and have correct written instructions before starting any job. Follow the instructions exactly as given to you. Always report errors and bad practices immediately. Ensure you have the right materials before you start a job. Use the correct equipment for the job, confirm its status and cleanliness. Maintain good segregation. Protect against contamination. Work accurately, precisely, and methodically. Maintain good standards of cleanliness and tidiness. Ensure that the changes are pre-approved through the change control system. Never assume anything. Before performing any task, check it out.
So as you can see, like this is the main basic 10 principles of GMP, but there's lots of other principles. It's like even if someone does any mistake, they are encouraged to come up and be honest and be transparent about that so that we can do the root cause investigation properly. So giving this training to new staff members who just come out of university is easy, but older staff who has been working in the institute for longer, it's a mammoth task to make them to follow GMP because university academia staff often don't want to follow too much rigorous paperwork because they always question, why do we have to record this? But trying to embed GMP, good behavior within them, GMP behavior, it's a task for quality assurance department. And we are often challenged and there are often debates as well. So we have to make sure that they listen to us and follow the procedures. So what is MHRA? So that's Medicines Healthcare Production Regulatory Agency within UK, and within the United States, there's FDA, and in India, there's another organization. Sorry, I can't remember the name of that at the moment. So these are the regulatory bodies which actually make sure that each and every pharmaceutical organization or a clinical trials manufacturing unit are following procedures and they often inspect us, sometimes with 1 month's notice, sometimes with 1 week notice. So we have to be audit ready all the time because inspectors can come anytime. And inspectors really are thorough. And when I did my auditing training, I was told to check the bin first, within a lab, because sometimes people try to hide data and they scrap some paper or raw data into the bin. And if that raw data is scrapped, that means that opens up a lot of can of worms and questions saying, oh, we are trying to hide data here. So then they start digging more and more. So within the COVID vaccine project, quality assurance started from quality by design into all experiments. So it started from beginning. When it came from R&D, the tech transfer team, when they transferred it within our organization, so we had to make sure that the tech transfer documents are followed properly and everything has been agreed and agreements are in place. And then there are raw materials which are used within the lab, for example, buffers, media, solutions, PCR equipment, everything. So we have to have a certificate of analysis, which also is handled by the quality assurance team who generates this certificate before anything can be used. And also we have batch records where we record everything for full traceability. Now, for example, the buffer components which are used in our vaccine production are tested for identification and also made sure that they are meeting the regulatory standards. So every staff has to make sure that they follow this procedure and make sure they have followed the standards and submit the documentation to quality assurance before going ahead with the next step. Then comes the batch review record review process. It's a multi-step process. First, the operator fills in the system, and then it comes to the head of production to check the documentation, and then finally it comes to quality assurance team. So it's a multi-step checking so that nothing is missed. We are making sure we are checking on every step. So after the quality assurance manager, which is me, I check every document, and then I give it to the qualified person who is the last person on the supply chain who certifies the batch. Now, what is a qualified person? So qualified person is a quality assurance professional mainly who gets certified by the regulatory bodies, and Royal Society of Chemistry and Biology in London has interviews them before they can pass the test, which currently I am undergoing at the moment. So for that, I have to study 12 modules like a master's course with my full-time job, doing that in evenings and weekends, and going through each and every modules. And then not only theoretical knowledge, I have to apply them in every day in my job to make sure that I'm learning properly. So I have my QP sponsor and supervisor within the university who guide me every day. So for example, today also before this presentation, I was doing some investigation research, one of the problems which had happened recently. So a qualified person has the authority of signing a batch before it goes to clinical trial, for trials products, and for final products before it goes to the market. For example, paracetamol, which goes into the market, is certified by a qualified professional. So they sign the final declaration, they have the legal responsibilities and duties, and they are always providing quality advice and quality coaching to all the staff within the organization. It is a very stressful job and a responsible job. If something goes wrong with the, for example, paracetamol batch has been certified by PQP and has gone in the market, and then one of the vendors or one of the pharmacies come back with a complaint saying, well, this has adverse reaction on a patient, then if it is a critical adverse reaction, then QP might be even go to prison because that person hasn't checked properly. That's why it has gone out to the market. So that's why it is a very responsible job and really stressful as well. So when a biotechnology production plant, how does it look like? I've been to many production plants in my career. I have worked in GSK, where we were manufacturing inhalers and different dosage, oral solid dose tablets like paracetamol. But traditional pharmaceutical plant is different than biologics, because biotechnology is a very evolving subject, as you all know, and every day changes are happening. And manufacturing a vaccine is a very complex process. And within that complex process, to make sure we are following GMP is a big challenge. So when I had my training for one of the qualified professional course, one of my teachers showed me how a large manufacturing plant looks like. I have been on site to visit one, but because it's not possible for people to go and see very easily, so I thought to share a video of a large biotechnology production plant in Switzerland and show how they work within the organization. And also it gives a bit of flavor to students to be motivated to pursue a career within biotechnology.
Only a few decades ago, the idea of harnessing biotechnology to produce medicines appeared highly fanciful. That's because unlike conventional drug ingredients, which are produced by purely chemical methods, biotechnologically produced medicines are much more complex and difficult to manufacture. Today, these drugs have become must-haves in the treatment of several serious diseases. Biotechnologically produced treatment tools are now available in several disease areas, cancer in particular, representing a beacon of hope for thousands of patients. Roche is the world #1 in biotechnology. With its research, development and production sites scattered across the globe, the Roche Group offers doctors and patients alike a vast range of biopharmaceuticals and diagnostic tests manufactured using biotechnology. One biotechnology production site is located at group headquarters in Basel. Beginning in 2007, after just 2 years building works, the facilities were steadily validated and accredited. Total investment ran to 400 million Swiss francs, or $450 million US. Production was launched in mid-2009. The production building being used for monoclonal antibody production was completed in just 33 months. The facility covers 10 floors and represents state-of-the-art biotechnology. The upper floors are used to prepare solutions for production. The facility is highly automated. Each biopharmaceutical product is unique. The slightest alteration in a single one of the many production steps can have an impact on quality. It all starts here, when a staff member takes frozen animal cells out of liquid nitrogen. The cells are the actual producers of the active ingredient. The cells are transferred to a nutrient solution and then propagated in stainless steel containers or bioreactors. The cells are sensitive and react to even the slightest of changes in conditions. It is therefore essential to strictly control and document this entire process. Samples are taken at regular intervals, poured into bottles, and then tested in the lab. Using special equipment, staff quickly analyze the composition of the solution. If the samples comply, the solution can advance to the next stage. The cells are gradually transferred to ever larger bioreactors. This process always involves critical examination of the samples. As the larger the reaction vessel, the more difficult it is to create identical conditions for all cells.
Amazing.
Monitors are located on each floor to ensure close control of the facility. Staff can also look inside the bioreactors through sight glasses. Sterile flexible hoses are used to transfer nutrient solution components, such as sugar. Under hermetically sealed conditions, these hoses are cut and connected using a special welding machine. Inside the 12,500-litre bioreactors, the cells grow and release the therapeutic protein into the nutrient solution. Actual product synthesis is then complete. Purification of the mixture can begin. Staff at the control stations check every step of the process. The cells are now separated from the solution by centrifugation and filtration. The precious solution is collected in harvest tanks. The next stages in the process require huge volumes of purified water, distilled to the highest possible degree of purity on the 2 basement floors that house the complete infrastructure. This is also where the purest grade of steam is produced, along with other process gases. High-performance pumps convey water and coolant to the production area on the upper floors. Pressure, temperature and other detectors continuously monitor facility status. Valve status also has to be monitored, only at longer intervals. After weighing out salts into special metal drums, the staff now prepare the system for filling. They use a crane to maneuver the drums to the vessel's filling port. This keeps the system hermetically sealed throughout each process step, including during the production of salt solutions, for example. After the fermentation and cell residue filtration phases, the product is purified. The staff convene briefly to confirm everything is okay. The salt solutions needed for purifying the product are then loaded with the harvested cell-free solution onto chromatography columns. Automatic measuring probes monitor the solution composition. They have to be regularly cleaned and inspected. Although the processes are highly automated, seamless staff communication and collaboration is absolutely essential. The product passes through various purification stages. The product exiting the last chromatography column is the pure protein monoclonal antibody. The purified product is filled into cryovessels and frozen ready for forwarding to the formulation department, where it is processed into the finished drug. Roche aims to develop innovative biopharmaceuticals and protein-based constituents for highly specific diagnostic tests, and to make these products available to patients and healthcare professionals worldwide.
So I wanted to show this because currently our COVID vaccine, which is manufactured in our small unit within the university, has been already transferred to AstraZeneca for mass production within UK. And in India, Symbiosis Pune is also manufacturing our vaccines in advance before the trials pass. So everybody in the world is looking forward to this vaccine. And as soon as the clinical trials pass, which is we are not sure yet because we have reached the clinical trial phase 3 at the moment, which is being trialed all over the world. So we are shipping our vaccines all over the world for trials, because a vaccine cannot only be trialed only on specific population. It has to be trialed all over the world because everyone's immune response is different. Ethnic minority people like us, our immune response is different. And if you might have seen in the news that lots of ethnic minority people, doctors and nurses have passed away in this pandemic all over the world and also in UK. So every race, their genetic composition is different and their immune response is different. That's why I wanted to show this video to see that how a large-scale manufacturing plant actually produces biotechnology products. And at the moment, lots of manufacturing are happening all over the world. So hopefully, if the trials pass, then the vaccine will be available to the common people in the market for saving our lives and making us immune from this deadly disease. Now, I thought to touch a bit on my journey because people always ask, how did you reach there? What have you done? So I started BTech in biotechnology from Heritage Institute of Technology in Kolkata. And then I decided to pursue a master's in University of Leeds in UK, where I have worked in a completely different project where I was looking at the intervertebral disc replacements in spinal cord injuries. And I was working with cobalt chromium metal nanoparticles in an in vitro model with porcine cells to see how it impacts porcine tissues and whether they grow with this implant. And after that, I was questioning myself, do I want to do PhD, follow the academia path as my father did? But I wanted to really work in the industry because I was more proactive about industrial experience than academia. So after lots of job applications, finally I got a job in GSK as an associate scientist within R&D facility in Hertfordshire. There I worked with scientists from all over the world, lots of different experience, and it's a pharmaceutical giant, as we all know. So I have learned a lot there in different departments, in quality control, in quality assurance, and it has been a life-changing experience for me. But I was impatient. I wanted to come out from a big firm and work in a startup. So I worked in Imperial College in London as a pharmaceutical quality manager, developing a quality management from scratch and also implementing ISO 17025 laboratory for quality control testing. But it was a startup, so the project ended within 6 months. So I moved to DITCOT in an active pharmaceutical ingredient manufacturing facility, worked as a quality assurance specialist there. And then last year, I moved to University of Oxford as a quality assurance manager. And currently, I'm undergoing qualified professional training, which is under certification of Royal Society of Biology in London. And this is certified by Royal Society of Chemistry and Biology both. And one of my current projects is a very challenging one, is implementing electronic quality management system within our university facility, basically transferring the paper-based system to a software system. Because during pandemic, everyone is wanting to work from home. So this will facilitate a lot of people to help to work from home rather than going on site. Just a couple of snapshots from newspaper cuttings, and Catherine Green, the head of our department, acknowledging my work. And current vaccine project, there's lots of things on media at the moment. If you're more interested, want to know, you can Google on YouTube about COVID vaccine, Oxford University. And there's some links which are on BBC News website as well. And finally, I would thank St. Xavier's College to giving me this opportunity to share my experience on the COVID vaccine project. And I'm happy to take any questions.
Thank you, ma'am, for your very informative and insightful presentation. Now, I would request Professor Ananita Banerjee to conduct the Q&A session as well as give you the formal vote of thanks.
Thank you, Moitrey. Thank you, ma'am. This has been a very wonderful lecture for the COVID-19, the current scenario which we are all facing now. And your lecture has enriched our knowledge very much. Now, with this, I would like to invite questions. The questions are already there in the chat box. So as you can see, the students have found this lecture very interesting, and the chat box is flooded with questions. Now, it's already flooded with questions, so I'll be trying to address most of them. So I'll be starting with the first one. So the first question is, doesn't introducing the spike protein in a harmless virus makes the harmless virus infectious? Do I repeat the question, or is it okay?
No, it doesn't make it infectious. And to be honest, I'm not the best person to answer this question because I'm not a virologist. So my experience is more on the quality assurance side of things. But I can take this question and I can get back to you asking my subject matter experts within my organization.
Okay, ma'am. So we move towards the second question. It says, from the recent news in India, there will be the use of COVID-19 vaccines. So according to you, madam, how will it be effective to the vast population of India? It has come from a student.
So you mean the vaccine to be effective within the vast population?
Yes. Because we have the different strains of viruses. So how a single vaccine will be effective against all these strains?
Yeah, so currently the clinical phase 3 trial is going on. So the trial data, until it comes live, we cannot 100% comment on that topic. But so far, the trial results have been really good. So we are 80% confident that this will work on different strains of the virus. And looking at the population of India, obviously social distancing measures have to be followed. Otherwise, until the vaccine comes into place, nothing can be done, to be honest.
Another question is, which is a very common question, because we know that since this is an RNA virus, so it is mutating a lot. So after the development of this vaccine, will it be effective to cure the virus? Because since this virus is an RNA virus, it is mutating. So then also this vaccine will be effective.
So at the moment, all over the globe, there are 8 vaccine candidates which are different. So ours is an adenoviral vector vaccine, but Moderna in US, they are doing the RNA virus vaccine. So I think it depends again. Not only we cannot only hold our hopes to one vaccine. So there are multiple vaccines will need to be live, and also they need to pass the clinical trial. And after that, they need to be produced in mass scale so that the whole world gets them. So that's why it's a very contradictory situation at the moment.
Yes, indeed it is. So another question is, a latest study in Cell showed that the spike protein underwent D614G mutation, making COV2 more infectious and globally predominant. Doesn't it put the efficiency of Oxford vaccine into question?
I'm not aware of this study, so I cannot comment on this. Sorry.
Okay. And then we go for another one. So it says, how do you identify and select a potential drug from a plethora of available pre-existing drugs?
So that goes through a screening process. So firstly, in the bioinformatics department is involved in screening the drug molecule to get it from the molecule structure to the R&D phase. And then the R&D department does the experiments and then confirms whether the drug molecule is potential to go ahead.
And is there any relationship between the climatic conditions and working of vaccine?
No, this hasn't been proven any climatic condition because initially it was thought that in hot temperature the virus doesn't work, but now like India is really hot and the virus is spreading. So climate doesn't have any impact.
What are the proper ethical guidelines to be maintained strictly before making a vaccine by and large available at the market?
Sorry, I didn't understand this question.
And do I repeat it once more? That is, it says, what are the proper ethical guidelines which needs to be followed before a vaccine is made available at the market?
Oh, before the vaccine is made available in the market, ethical guidelines would be for normal human beings in the out there to maintain, wear a mask, use sanitizer, maintain 2 meters social distancing, do not visit family and friends, just go to work and be in your home. That's what I'm doing at the moment.
It requires a lot of sacrifice.
Yes, that's why everyone in my department is really stressed because we do not have anything else apart from work in our lives at the moment because we have been strictly told do not get sick and ill because we need you.
Yes, the whole world is looking towards you. We also need you. So take good care of yourselves. So
Thank
the
you.
next, so the next question is, what is the scope of person from biochemistry background? So this is from a student indeed. He wants to know his future career prospects. So what is the
Yes.
scope of person from biochemistry background in quality assurance department after masters?
So after masters, if you've done a masters in biochemistry, you can work in quality assurance department. But initially, I would say that if someone gets an experience in R&D research or quality control, then it's easier to move to quality assurance. And also, it's like what type of job you apply for. If you want to go for an entry-level job, then quality assurance sometimes accepts application depends on the product they are working on. So if it's a, for example, active pharmaceutical ingredient, which involves more chemistry, then obviously they would be interested in hiring someone from a biochemistry background.
And it follows another, this question is followed by another question, that is, what are the factors that a person should look for before choosing a quality assurance company for applying? Before
For
he wants
applying
to apply
what?
for applying for a quality control job, I guess.
Job. Oh, okay. So mm, so mainly for doing a quality control job, you need to have basic scientific background. So if you have a master's in biochemistry and you can prove yourself in your interview that you're competent enough to learn new things fast and apply them, a manager or someone hiring would be interested to hire you basically. So it doesn't need to be like I've done X, X, Y, Y, I need to get the job. It's not very strict measure to hire because as you know, biotechnology and all these fields are evolving every day. So we are looking into hire new people with new fresh ideas. So if someone proves themselves in the interview, they are competent to do the job, they are often hired.
And the last question is, different country have different types of education system. If a person from developing country apply to developed country, then how the equivalency of the degree will be met?
So for example, myself, when I went to study masters, my equivalent degree was evaluated with the standards they had, and it met their criteria. So anyone from a developing country, for example, my scores used to be in GPA, and UK has their own systems. But when I sent my transcripts through, they compared to their own evaluation and they accepted my application. So it's not really difficult to go from a not developed country to a developing country to study.
Thank you, ma'am. This brings to the end of our question answer session. So I hope I have met all of the queries so far. So now it is my pleasant duty to offer a vote of thanks to the Honorable Speaker, Ms. Chandrabali Doctor. She has taken out a valuable time amid this busy schedule to address us. We are extremely fortunate to have a front runner in the fight against COVID-19, and she has explained very well to us how the battle is being fought. She has described all the challenges that she is being faced and addressed. She sailed us through a beautiful, lucid, and videographic journey starting from origin of vaccines, the thumb rules of good manufacturing practices, the role of regulatory bodies such as MHRA in keeping the quality control of vaccines, the current COVID-19 vaccines projects, which are being under in different phases of clinical trial worldwide. And also she has discussed the different personal experience that she went through. I hope that this lecture has been immensely helpful to the students, and the whole lecture, I know the content of the lecture is a part of their curriculum too. So with this new information, I hope that we will be able to find a remedy to this virulent pandemic which has devastated normal life across the globe. All the best to your endeavors, ma'am. Thank you.
Thank you very much for your kind words.
Now over to Moitri.
Thank you, Dr. Anindita Banerjee, and thank you, Ms. Chandrabali Datta, for your beautiful presentation. We are at the concluding session of this international webinar, and I would request Organizing Secretary, Assistant Professor, Dr. Arup Kumar Mitra, to give the concluding speech. Sir, over to you.
Thank you very much, Moitri. Now, every good thing has to end. So FIBS 2020 is also ending, and it is my duty to narrate what has happened in this new type of exercise which we have initiated. Let me tell you, at the outset, I was not really confident that whether we will be able to have this webinar without much difficulty. But somehow it happened with illuminating talks by different speakers. And of course, at the forefront, we had our father principal who gave a very illuminating and at the same time confident talk of meeting this new normal situation and what we should do really to have ourselves on our toes in this condition. And the basic topic of this particular webinar was basically the COVID-19, the current scenario. So all the speakers spoke in this line. Dr. Ranadhi Chakraborty spoke on the virus itself, the cell cycle, and the different ways of its infectivity and how dangerous it is. Then we had this afternoon session from Dr. Anindita Banerjee, who benefits of human psyche in this COVID-19 and the lockdown period, and imparted great deal of positivity in the minds of the listeners. And of course, each of these sessions on the first day was followed by active question answer session. Then we had the second session with Dr. Shashirekha Mundra and Chandrabali Datta, who actually spoke in different aspects. Dr. Mundra explained the immunological, epidemiological, and structural point of view of COVID-19. And of course, she also talked about yoga and meditation and its scientific explanation, how it actually improves our gut microbiota and help us to survive in a better way. It was really an interesting and illuminating talk, and we all enjoyed it. And the last lecture by Chandrabali was really wonderful. It is not only because of the fact that she is one of the forerunner against this COVID-19 virus, it is also because of the fact the way she presented it. It was actually felt by me that through this particular lecture, through those videos, the students have learned a great deal. I was really very interested to see the way the biotechnology-based products are developed in industry and how really these products are formed without human interface. I really thank all the illuminating speakers, and I feel that the students have gained the speakers' knowledge and the overall ambience of this webinar, and I strongly believe in future also we have actual lecture in the form of seminar, but nevertheless this webinar has made certain standards. Thank you very much.
Now, I would request Dr. Oyun Chandra, Member Secretary of Academic Council, to give his final vote of thanks. So please.
So we are at the end of a very enriching webinar of 2 days. It is my present duty to thank a few persons without whom this wouldn't have been possible. First, I'd like to thank our speakers, Dr. Ranadhir Chakraborty, Dr. Anuttama Banerjee, Dr. Shoshirekha Bundra, and Dr. Chandrabali Dutta, who are stalwarts in their respective fields. I'd like to express my gratitude to our beloved Father Principal, Reverend Dr. Dominic Savio, whose inspiration was always there with us. I'd also like to put on record my words of gratitude to the members of Advisory Committee, Professor Bartram Da Silva, Vice Principal, Arts and Science, Dr. Tapani Datta, Dean of Science, Dr. Argho Banerjee, Dean of Arts, Dr. Parthopratim Ghosh, IQAC Coordinator, and Dr. Subeshna Sham Chaudhuri, the Head of the Department of Microbiology. I'd like to mention specially about Dr. Arup Kumar Mitro, the Organizing Secretary, as well as the Organizing Committee members, Dr. Kosturi Sarkar, Ms. Debjani Datta, Dr. Mahashweta Mitra Ghosh, Dr. Madhumita Moitro, Dr. Riddhi Mojumdar, Dr. Joydeep Ghosh, Dr. Anindita Banerjee, and Mrs. Lopamudra Roy. And last but not the least, there are a bunch of students who have made this webinar possible technologically, and very smooth organization was done by them. I'd like to mention the names of a few of them. They are Madhura Mondol, Moitri Sarkar, Tanishta Bishwas, Srijan Bhattacharya, and Swarnali Chatterjee. Thank you, everybody. May God bless all of us. Thank you.
Thank you, sir. This marks the end of the two-day international seminar, Frontiers in Biological Sciences 2020, Chapter 4. And over to my participants, I would like to inform everyone that there is no feedback link and there is no feedback form as well. We'll be going thoroughly through the YouTube analytics to mark your attendance, and based on that result, e-certificates will be produced. Production of e-certificates will take around 20 working days, so please keep your patience. And also, we would like to sincerely apologize for any sort of inconvenience caused. And thank you once again, and good night and take care. Bye-bye.